But okay, I think let's see. I like this. Let me, I guess we're getting near a quorum, so I think this is okay. Okay, I'd like to thank Tony. Introduce our speaker, Tony Zador. He is at Cold Spring Harbor. He is a computational neuroscientist who has had a long interest in AI and machine learning. He has an MD, PhD from Yale, okay, which I think I've heard of, and he's going to talk today, I guess, about a critique of pure learning. And I think I'll
All
turn
right,
it
well,
to Tony.
great. Well, thanks for the introduction. I should actually add that I wear several hats. I'm not only a computational neuroscientist, but my day job really is to run an experimental lab. And a lot of my experimental lab is focused on figuring out how to decipher neural circuits in behaving animals. And one of the things that we do is we grind up brains and use high throughput DNA sequencing to determine the circuits of those brains. But that's not actually what I'm going to talk about today. I just wanted to point that out. Indeed, what I'm going to talk about today is a critique of pure learning and some thoughts I have on basically how AI can benefit from taking more inspiration from neuroscience. And if I can, there we go. So, our goal in some sense is to build machines with general intelligence. I'm not exactly sure that I like the term general intelligence, nor would I want to define it. But, deep down, I think my inspiration is maybe shared by many people in the field, which is, I grew up watching lots of science fiction, and there are lots of robots and cognizant artificial entities in these movies, and it remains frustrating to me that they're still only in movies. I figured, when I was a kid, I figured that the real challenge was going to be that by the time I got to be a grown-up, the problem would be solved and there'd be nothing left for me to do. And that turns out not to be quite true. So this is a walk down science fiction memory lane. If this were a live audience, I would invite people to see if they can identify all 6 of these. But if you want to put it in the chat, by the way, I would love interruptions. So I invite people to ask questions or make comments. My nightmare scenario is something I read about on Twitter recently, where somebody started a talk and then a few minutes in, he started getting bombarded with emails, and then he shut down his emails, only to discover later that those emails were his host telling him that his Zoom connection had died, and he had spent 45 minutes talking into a camera with no one on the other side. Anyway,
No, we can hear you.
Okay, well, I appreciate that. Anyway, if anyone wants to throw in the chat what these are, I'm all for it. Anyway, the question is, why aren't we there yet, and how do we get there? So as many of you know, the old school, I'm old enough to remember, in fact, I started graduate school when the traditional technique was good old-fashioned AI. And in good old-fashioned AI, which one might refer to as sapiocentric, human-centric AI, you focus on the things that seem most uniquely human, like chess and language and stuff like that, because that's obviously the most important thing. That's obviously the hardest part. And then you sort of go out to the things that are maybe a little bit more less challenging, less interesting stuff that maybe we share with a few hundred maybe creatures on Earth, like planning and reasoning, octopuses that can solve the Rubik's Cube, monkeys that can use tools. And the boring stuff is the stuff that all animals do, right? This is the traditional view of AI. But I'm always inspired by what's known as Moravec's paradox, who put it so nicely, so I'll read you the quote. He wrote, "Encoded in the large, highly evolved sensory and motor portions of the human brain is a billion years of experience about the nature of the world and how to survive in it. the deliberate process we call reasoning is, I believe, the thinnest veneer of human thought, effective only because it is supported by this much older and much more powerful, though usually unconscious sensory motor knowledge. We're all prodigious Olympians in perceptual and motor areas so good that we make the difficult look easy. Abstract thought, though, is a new trick, perhaps less than 100,000 years old. We have not yet mastered it. It is not all that intrinsically difficult. It just seems so when we do it. So in other words, the hard part isn't playing chess or even having a conversation. The hard part is interacting with the world. What animals do is hard. So, in the same way that there was once a Copernican revolution that moved the sun from the Earth from the center of the universe to putting the sun at its rightful place, so we should have a Copernican or Moraveckian revolution that places animals at the center, and the stuff that we do, the uniquely human stuff, as sort of almost a byproduct. So that's what that's my thesis here. I'm gonna try to talk about both why I believe this to be true and some work we started doing toward pursuing this idea. So our goal is to achieve mouse level AI, because mouse level AI would provide a solid foundation for some of this other stuff we might be interested in doing. And I'll just mention that we have a Neuro AI Scholars Program at Cold Spring Harbor for people with a PhD in AI, not a traditional postdoc, and it focuses on applying insights from neuro to AI. Scholars are embedded in Cold Spring Harbor for 2 years, and they're independent and expected to forge new paths. So here's the outline of my talk. The first point I'm going to make is that AI relies mainly on learning, whereas animals rely on innate structure. And I'm going to argue that animals are no better at learning than our machines. In fact, machines are probably at this point better at learning, and that what sets animals apart is not their ability to learn in their lifetime, but what they've acquired through their genome. And what they acquired through their genome is expressed through development in the innate structure that is embedded in their wiring diagram. And the wiring diagram is compressed through a genomic bottleneck. And so I'm going to talk a little bit at the end about the genomic bottleneck algorithm that I've been working on with Alex Kulikov. Okay, so as many of you know, as most of you know, modern AI relies on learning. In fact, nowadays, AI is often used synonymously with machine learning. So here's a sort of classic machine learning task. You take a lot of labeled pictures, use gradient descent, backpropagation, and you convert, you squeeze out the structure in those pictures, and you embed it in the weight matrix of, let's say, feed-forward network. You can have n layers with whatever structure you want, but basically, what learning does in this model is it squeezes the structure of natural images into the weights, the synaptic weights of a weight matrix. So in this traditional approach, neural networks start tabula rasa. They start with just some random initial conditions, and you need a lot of labeled examples to learn all these parameters. So we know that humans do not rely mostly on supervised learning. Labeling every object every second, labeling objects every second for a year would still only be 3 times 10 to the 7 training examples, which is, depending on the particular test, still orders of magnitude away from what artificial neural networks do. And of course, we don't label objects for our children every second for 2 or 3 years, much as it might seem we do if you have a young child at home who's asking why, what is that? So traditionally, the resolution of that. So this mismatch between how humans and even animals learn and the number of labeled examples, the sort of neediness, the greediness of supervised learning algorithms for data has been recognized for decades. And the usual answer to that when I was a kid was unsupervised learning. And in fact, right, because for unsupervised learning, you get a lot of data, let's say, from your sensory apparatus, but it doesn't require the label. So back in 2016, Jan LeCun put it this way. He said, if intelligence is the cake, then the bulk of the cake is unsupervised learning. The icing on the cake is supervised learning, and the cherry on top is reinforcement learning. So that's his idea, that basically the bulk of what we do is unsupervised learning, and these days he sort of tweaked that, and now he refers to so-called self-supervised learning. But I think it's pretty clear that even unsupervised learning isn't enough for many of the things that animals do. So a squirrel can learn to jump from tree to tree in a matter of weeks after birth, maybe months. Horses can stand within seconds after birth. Spiders basically hunt out of the egg ready to go. So most animals are basically, they spin up just ready to go. They don't need to be squeezing a lot of structure out of their environment in most cases. So, even more compelling are behaviors that animals can't possibly learn. So termites build termite mounds without instruction from other termites. That's somehow an innate behavior. Birds build nests same way. Beavers build dams. Now, you might think that these are all learned behaviors, but in many cases, we know that learning is either completely unnecessary or not a major component. There's a compelling YouTube video that you can go online and watch. Just search up Justin Beaver, the rescue beaver. So some woman found a beaver pup. I don't know what a young beaver is called, and raised it from a very young age. And at this point, it's now an adolescent or older. And she documents all the ways in which it behaves in beaver-like ways. Basically, it spends its time trying to build dams in her house. So like JB took my daughter's lacrosse stick one day, and it starts gathering up all these little pieces, starts gnawing at the doorstops, trying to cut them down like a tree to build a dam, and then starts putting them into, dragging them around the house and building these little dams, especially near the bathtub. So this is like somehow built into Justin Beaver to build dams. This has been studied in detail by Hopi Hoekstra, who's at Harvard. She studies two related species of Paramiscus, which are field mice. There's the old field mouse, which builds these so-called escape tunnels. So it's a deep tunnel, and then there's a little escape hatch with a tiny bit of dirt here on the top. And the idea is that if some critter comes in chasing the field mouse, it can run up this way and pop that little bit of dirt out and escape going out that way. But interestingly, deer mice, which are so close they can actually interbreed, don't produce those escape tunnels. They produce, they build stubby little tunnels that look like this. And she knows that this is encoded in the genome through cross-rearing experiments, where she takes pups like this that have never been exposed to their biological parents, and they're reared by foster parents of this sort, and vice versa. And it turns out that the tunnel building behavior of these mice follows their biological and not their foster parents. So why do we have innate structure like this? Well, I mean, basically, if you think about evolution, evolution is all about a race to produce progeny. And you are at a disadvantage if you come into this world knowing nothing, all right? So if you come into this world pretty much ready to go intellectually, you only need to learn a little bit. I'll get back to what one should learn, but if you only need to learn a little bit, then you're likely to survive. If you start off as tabula rasa AI systems do, if a biological organism were to start off as a tabula rasa AI system does, then it would be helpless at birth, as humans are. And it would take a very long time to spin up and get to the point where it could move forward. So from an evolutionary perspective, there's an obvious and overwhelming advantage to building in as much innate knowledge of the world as possible. And I'll say as possible, but there is a limit, right? You don't want to build in too much. You don't want to crystallize things that are highly sort of variable in the world. So there is a trade-off there, but it's way more than traditional tabula rasa AI systems. So evolution will typically maximize fitness at birth by incorporating innate structure. I'll just give a couple examples here of another important contribution or sort of role of innate structure, which is they speed up learning. So I don't have time to go through this very nice paper, but basically there's an area of the mammalian brain called the hippocampus that's been well studied in rats and mice. And that area of the brain encodes a map of visual, of space, not visual space, of space. And what's interesting is that this map is very flexible because animals move from environment to environment. So you put a mouse or a rat into a new environment, and within a few minutes, you have neurons whose firing rate, that's what these bright things here is, the firing rate is a function of position in an environment. So these are, you get neurons whose firing rate is dedicated to particular positions. But the ability to acquire maps of the visual space, of the physical space in which an animal finds itself in, that's built in. So it's basically like you have all the structure you need to know how to build a map, and then the creature, as soon as it's put into an environment, acquires the map of the place it's in. This is one of the most beautiful examples of that. This is work from Konrad Lorenz, who discovered that geese imprint early on who their mother is. So if what he did was he took very young geese, and I guess I'm not exactly sure what he did with the moms, but he replaced himself as a surrogate mom. And the young geese came to the conclusion that he was their mother, and they followed him around throughout their lives like a mother. So this sort of starts letting scientists get a handle on what's built in and what's learned from the environment. What's built in is the ability to or the need to imprint upon a particular parent, but the details of that parent obviously aren't, because he doesn't look exactly like a goose. I'll skip over some of these others, just say that humans probably have innate priors that enable learning language. And if you want to read more about what animals can do, there's a wonderful and very readable book by a primatologist named Frans de Waal called "Are We Smart Enough to Know How Smart Animals Are?" And I highly recommend that to anyone who's interested in what animals can and can't do. And I'll make a minor point, side point, which is that animals aren't even that good at learning. So as I said, a lot of my day job is as an experimentalist. And I spent the first few years of my time at Cold Spring Harbor training rats to perform what seems like a very simple task. So the task is basically to train the rat to stick its nose into the center port, and then that elicits a sound. And then depending on whether the sound is high or low frequency, the rat goes left or right. And that, I've just explained to you in a couple of sentences how to perform this task, but to get rats to perform this task can take, well, we ended up getting better at it, but when we first started training rats, it took the better part of a month or two to train them, of daily training Now we can get it down to about a week. I should say that the rats are highly motivated to do this because each time they do it successfully, they get a little water reward and they're thirsty, so they're highly motivated for that. And even monkeys, people who work on awake behaving monkeys similarly find that it can take months and months to train the monkeys to do the basic paradigms that they would like them to learn. So training requires careful shaping, adapting the task to what animals do innately. That's actually an important point, that in the end, we tried several different ways of training these rats. And the reason we were eventually able to get them to learn this task pretty fast is we exploited, for example, a rat's innate tendency to explore little holes by sticking their nose in it. Okay, so the conclusion from all this is that the brain comes mostly pre-packaged, that this view is wrong. This is not how it really goes. Really, the brain comes in a package and you just add water, throw it in the oven, and in a couple weeks, you get an animal that, or a couple of days, sometimes even at birth, you get an animal that's ready to go. So here's the summary so far. Artificial intelligence relies on learning, and by contrast, natural intelligence relies on innate strategies. Okay, so now I'm going to go to part two, which is, well, where did these innate strategies come from? So basically, everything that is needed to spin up an animal is contained in that animal's genetic code, its DNA. Innate behaviors are, one way or another, indirectly encoded in, hold on one second, I'm getting this flurry of text. Wish I knew how to turn it off. Yeah. I was hopefully not texting that you guys have all dropped off. I can still see you.
Oh, we're still here. We're still here.
Yeah, I can see you. So innate behaviors are encoded in the genome. So just to remind people who aren't biologists that everything, the genome encodes pretty much everything that's inherited by an individual, right? So there's often sort of fuzziness about, well, maybe there's extra stuff that's not in the genome. Nope, it's all in the DNA. And we know that because we can, for example, take the DNA from a black sheep and put it into the nucleus of a different sheep, right? This is how they made Dolly the sheep a couple of decades ago. And so if you take this DNA here, put it into that DNA there, you end up with a black sheep, not a white sheep, not a half white, half black sheep. And in principle, you can even do this across species. It's a little bit more challenging for technical reasons. But in principle, you could take, to first order, the way to think about it is you could take the DNA from, a human and put it into that egg, and you should get out a human, more or less. There are some minor details that we could quibble about. So that's only approximately true, but conceptually, that's about true. So what does the genome encode? Well, it encodes everything, right? So it encodes molecules, it encodes how synapses work, it encodes neurons. And we know a lot about all these parts, but most importantly, for the purposes of this, it indirectly, one way or another, encodes the wiring diagram of a brain. It encodes which neurons are connected to which neurons. That's, in fact, the sort of informationally, perhaps the most challenging thing it has to encode about a brain, the thing that sets the encoding of the nervous system apart from the encoding of other body parts. So how many parameters does it take to specify a brain? Well, so one of the simplest organisms with the nervous system around, and one of the simplest model organisms that neurobiologists study is called C. elegans, and that's a worm. It has 302 neurons and 7,000 synapses. All right, so and conveniently, people have figured out the exact wiring diagram of C. elegans. It turns out that it's highly stereotyped. So if you take two C. elegans and carve up their nervous systems, you'll find that almost all the connections are identical from one individual to the next. So is 7,000 synapses a lot or a little? Well, compared to what? Well, the C. elegans genome is about 100 million bits of information. So as you know, DNA is basically just a long string of letters, and there are 4 different letters, A, T, G, C. And so we can put an upper bound on the information capacity needed, that is stored in a genome. So if it's, 50, I say 100, about 100 million. So a little bit more complicated than that, but still, if it's something is 100 million nucleotides, and it takes 2 bits to specify each nucleotide, because there are 4 possible nucleotides, then that gives you a bound. So 100 million bits is more than enough to specify the 7,000 synapses connected to that connect up 302 neurons, right? So for this simple organism, every connection could in principle be explicitly specified in the genome, right? Because you would have a 302 by 302 sparse matrix, and it wouldn't take too much effort to specify each connection perfectly if you have, 100 million bits to go with. On the other hand, sorry, that should be a 10 to the 11th group, but on the other hand, a human brain has 10 to the 11 neurons, and it's sparsely connected, so it's about 10 to the 14 synapses. So it should potentially have 10 to the 11 squared or 10 to the 22 parameters or bits to specify. And that's vastly more. You can do that a little more carefully, and you end up with something more like 10 to the 15 bits to specify an arbitrary human brain. But that's vastly more than the human genome. That's 6 orders of magnitude more than what we have in the human genome. And that's if every single bit in the human genome were devoted to wiring up a brain, which it's not. So we have a missing factor of 10 to the 6, 10 to the 7, 10 to the 8 between what we have in the genome and what it would take to specify an arbitrary brain. So, this is kind of potentially a mystery. And I call that mismatch, i.e. the difference between what it would take to wire up an arbitrary brain and what we actually have available in our genome, the genomic bottleneck, right? There's an information bottleneck that prevents all the information needed to wire up an arbitrary brain from being available.
Can I ask a stupid question? Sorry.
Yeah, go ahead.
You're using basically 1 bit to represent an edge there. In a real brain, would the weights and stuff like that be much larger than the connectivity, or is it really the topology, the hard thing to get?
Yeah, so that's a really good point. It's a very interesting point. I think I didn't mention that here. Yeah, I sometimes have an extra slide on that. So first of all, real connections aren't just 0 or 1, right? You have a synaptic strength associated with them. You can do some estimates of how many bits are needed to specify the strength of the connection. In a lot of artificial neural networks, people use floats, and so that might be 32 bits or something. In a real brain, there's a lot of noise associated with the effective strength of the connection. And so, depending on exactly how you squint, you might say that it adds a few extra bits to each connection to specify not just the presence of that connection, but its magnitude. That said, so a lot of our intuitions for the amount of information it takes to wire up a network comes from thinking about small, densely connected networks. when you think about a large sparse network, most of the information is dedicated to figuring out who your partner is, and then there's only an extra couple bits specifying the strength of that connection. So I have that calculation spelled out somewhere, and I don't remember all the numbers off the top of my head, but basically the answer is for a network the size of the human brain, by far the majority of the brain. See, somebody wrote, there are other types of connections besides wiring. Nematodes have more GPCRs than we do. Yeah, that's true. I mean, we could go back to whether that qualitatively, this is a sort of back of the envelope calculation. I don't think that those sort of, I mean, I'm skipping over a lot of things, but I don't think any of them, and I'd be happy to discuss whether any of them qualitatively changes this argument. I'm going to go back here to the main thrust of the discussion. The genome specifies rules for wiring the brain. This is the basic resolution. The resolution is that we're not specifying, it's not like a long list where we say, you know, neuron 1 is connected to 3, 4, 18, 1,227,422. It's not an arbitrary list. There are rules, right? So we should be looking at really something like the Kolmogorov complexity, the length of the rule set needed to wiring up a brain, right? And you know, just to make this point more obviously, here you have on the left, well, yeah, you have a potentially arbitrary connection matrix, and that, you know, we'll use the full entropy of the, you know, whatever 302 squared parameters need to specify. On the right, I have like a trivial network where each neuron is connected to its 4 nearest neighbors. And the description length of that particular network is, you know, very, very short. Like I just gave it, connect to your 4 nearest neighbors. So the question is, can we discover the rules, or rather, are there simple rules that can capture a connectivity matrix that nonetheless enable that connectivity matrix to perform interesting computation, right? Because this thing on the right, although it has a very short description length, it probably doesn't compute anything terribly interesting, right? You need a few more degrees of freedom to allow the weight matrix to compute something interesting. And that's the idea of this genomic bottleneck that I'll be talking about. So here's the summary of part 2. AI relies on learning from examples. Natural intelligence relies on some kind of a genomic compression of W, right? You want a compressed representation of W that distills as much of the information that is useful in W to perform whatever task you need to perform. And so here I'm going to start talking about the algorithm. This is work done with Alex Kulikov largely, and now several other people in my lab. So basically, we attempted to take standard machine learning type problems, like bread and butter problems, and apply this genomic bottleneck approach to compressing the wiring diagram associated with these problems. So here, for example, is a simple artificial neural network that solves the digit recognition problem, the fruit fly of machine learning MNIST, right? So basically, again, this is an idealization. You might have more layers or whatever, but this is just an idealization. You have images coming in through the left. These little units would be pixels, for example. You present one of these images, and then you train it up with labeled examples. So the 10 written to should produce as an output the number 2. I guess this architecture is a little weird. I should probably have had 10 little outputs here, but I didn't feel like drawing that many lines. So the key point here is that the information, once you're done training this, all the information is embedded in this weight matrix W, right? And if it's multi-layer, then you just have to be a little more careful about how you write down W. So the goal in the genomic bottleneck algorithm is going to be to compress the weight matrix W while still maintaining good performance on the task, right? So we're going to try to squeeze the representation of W down as much as possible while still retaining as much as high a success rate in classifying digits as we can. And the compression is performed via what we call a genomic network or a genome. So yeah, so here's the sort of picture. We start off with a weight matrix that solves the problem. We compress it into into what we call a genomic network. And that genomic network should have a smaller description length, fewer parameters than the original weight matrix. And then we run this genomic network, and we come up with an estimate W star that is close to W, not necessarily in the sense of being close weight by weight, but rather in the sense of being close in that it produces similar outputs for as the original W, right? So here, let me talk a little bit more about how we go about doing that. Here's our weight matrix. This weight here, Wij, is the tells the coupling between some input neuron I and some output neuron x sub I and some output unit x sub j. Let's see, actually, sorry, I messed up the labels here. This should say P. This one on the right should say P network, and P there stands for phenotype, which is a biologist term for the thing that you actually get to observe. And this thing on the left should be a G network, genomic network. So the interesting thing here is the structure of the genomic network that we built, okay? So the idea is that we represent each unit of the, each pair of units in the, this is bothering me so much, I'm just gonna fix it. All right, there. Right. So each unit in the input to the G network is a pair of units whose weight we are trying to predict. Okay, so this genomic network is going to get as its input pairs of neurons from the phenotypic network, the P network. In fact, it gets an XI and an XJ. And it represents the XI and the XJ as sets of units. So each of these units is represented as a binary string. And now the genomic network takes that binary representation of a pair of units and tries to predict the coupling weight, Wij, that connects Xi and Xj. And this is sort of the central idea of how this genomic network works. It takes as its input a pair of neurons, and it produces as its output a prediction of the weight coupling that particular pair of neurons, right? And so this will work if this has the potential to work if these weights aren't all random, right? This will have the potential to work if there's structure in the W here that can be extracted by this genomic network. And in an image, for a network that solves an imaging problem, you might imagine that there is such structure, right? You might imagine, for example, that weights associated with nearby pixels might have some kind of characteristic behavior. that's the kind of structure that we can hope to extract in this G network. So the compression occurs via the G network. The input to G is a pair of neurons, and each neuron xi or xj is identified by a unique binary string. And so if there are n input neurons, then xi is represented by log-length string. Okay, and then basically we iterate this. The overall algorithm is we take an initial guess as to W, we train it just using standard stochastic gradient descent to produce an estimate of the weight matrix or to produce a weight matrix that actually solves MNIST or CFAR or whatever problem. And then we take that thing and we compress it down, and that produces an initial estimate of a better initial estimate of the weight matrix. And then we do that again. And we repeat this enough times so that we actually can get convergence to a compressed W initial, which has an extra T there, I see, which produces good performance without any training, right? So this basically, this is what you might imagine, this top line is what you might imagine an organism learns during its life. And this bottom one is sort of a representation of how evolution works. So how well can we do? It turns out that we do very, very well. So if we apply this to MNIST, we can, so a naive network should get performance of 0.1 because there are 10 digits. So if you guess randomly, that's what you do. The best possible performance on this task is, I think, about 0.95 or 6. That's this green line. And if we have 300 plus fold, 324 fold compression using the genomic network, we get performance that's almost indistinguishable from the full network. So in other words, we can compress the performance of a fully trained MNIST network by a factor of several hundred without damaging the performance. We can even go down to about 1,000 with only a very modest loss of performance. So what that's telling you is that the amount of information naively that you might think that is encoded in the weight matrix that solves MNIST is far, far less than the actual description length of those weights. We can do the same thing on CFAR, which is a more challenging data set using requiring labeling of images. And we get somewhat less performance, but it's still pretty substantial. You can get a 100-fold performance with very little loss of performance, and even at 400, you still get you get a substantial loss, but not a complete loss. So more complex network is less compressible, but still considerably compressible. I'm going to skip over this and just say that we've now moved on to reinforcement learning problems. And here's some classic reinforcement learning problems from, it's an Atari game that was solved by a really nice paper by DeepMind about, I guess, 8 years ago now. And without going into the details of it, there's a small reinforcement learning network that solves this. And I guess I should improve these figures, but basically, depending on the particular task, we can actually see excellent compression down to 1,000-fold compression or multi-thousand-fold compression. And actually, we have data on this. I just don't have the slide from it. We're now applying it to the OpenAI Gym learning of the half cheetah, where this cheetah has to learn. It's a simulated environment, and it's a pretty challenging task. It turns out this little stick figure cheetah has to learn to run, and it has all sorts of weird strategies, but the compression actually turns out to work extremely well for this. So that's about we have more results, but rather than going into those results, I'm going to sort of zoom out and sort of summarize what I'm saying, which is that really what we want to shift to, I think, is away from a traditional Turing test which seeks to mimic purely human behaviors like conversation or solving games or even solving math problems. And think about if we really want to get working on, a general kind of intelligence, we should think about an embodied Turing test. We should think about training organisms or training agents that solve what's actually hard, which is interacting with the world in a meaningful way, building dams, walking, swimming, flying, jumping from tree to tree, hunting, all those things. Those things are incredibly challenging, and we're not even close to being able to do most of them. So I'm just going to say a few philosophical words at the end. I started by saying that it's all about innate ability and not about learning. And a lot of people push back and say, well, okay, fine, maybe bees don't depend so much on learning, or maybe that's not what's so impressive about them. Maybe not even squirrels, but humans. We're all about the learning, right? We're useless for our first 5 or 10 or 20 years of life. Maybe we're useless until we get our PhD, and that's all about learning. So is learning why humans are so successful? I still say no to that. Sure, learning is important and we're pretty good at it, but that is not why we are so successful. If by successful we mean that's why humans have basically come to dominate the earth and out-compete all other animals. Although it's probably true that humans are very good at learning, it's not clear that we're that much better at learning than other apes. And I don't think that the learning is key to our success. And I'll point out that modern humans have been around for over 100,000 years. And actually, for most of that time, we were just an irrelevant afterthought to the inventory of organisms on this planet. If an alien came down to the planet 50,000 years ago and took a survey of life on Earth, and they said, okay, which are the really successful organisms? Humans would not have cracked the top 100 list. Back then, our overall population was measured, I think, in the 5 or tens of thousands. We hadn't really done much, and yet we had the same brains. So what changed? Why is it that for most of our time on Earth, we had no impact? We were not successful in any meaningful way, and suddenly we exploded in our success. Well, really, what made us successful was that we figured out how to start accumulating information from generation to generation. We figured out how to break the genomic bottleneck. And the way we did that was initially through oral transmission. We told stories. We instructed our young on how to do farming, how to farm, how to build tools. We bumped into other groups that figured out cool farming tricks, and we borrowed and stole those ideas. And then that process really accelerated as we went from oral transmission, which is higher bandwidth from generation to generation than just a genome, and is more flexible than just the genome. But it really accelerated when we went to written transmission, when we started passing information along in books. And now that we have computers, it's even more impressive. But it's true that some of our uniquely human abilities, specifically language, are a prerequisite for this transmission. But I think that the key to our success is not learning and not even language per se. It's the accumulation of information across generations that is enabled by oral and written transmission. So in summary, AI relies mainly on learning. Animals rely on innate structure, and animal learning, I argue, is no better than machine learning. Innate structure is involved taking, is embedded in the genome and is expressed through the wiring diagram. And the wiring diagram is compressed through a genomic bottleneck, which we can actually exploit to make better algorithms. And that's something that we're pursuing pretty actively right now. And I always close with this wise fortune cookie. If the brain were so simple we could understand it, we would be so simple we couldn't. And let me just thank, on this one, I really like to thank my collaboration with Alex Kulikov and my many funding sources. And thank you for your attention.
Okay, that was very interesting. Do we have some questions? Give this another minute or two.
Somebody raised their hand.
Someone raised their hand? Okay, I would say yell if you rose your hand. Who rose your hand? Anthony. Oh, can you unmute yourself? He may not be able.
Not possible. Maybe.
Not possible. Okay, he's got magic now. Fine. Okay, Anthony. Now try now.
I think maybe you have to unclick the mute on him, whoever is the host. Oh, maybe I'm a host.
We are both hosts, so.
Okay, I think I just clicked on ask to unmute more.
Disabled. Okay. Sorry about that.
Is there, okay, I'll just.
Actually, let's try typing it on the chat and we can read that.
Yeah.
Just type your questions on the chat.
Is there any biological evidence of how actually genome encode compress all the wiring of our brain? Yeah, I mean, the field of how you go from genes to a wiring diagram, biologists study that. It's called neurodevelopment. We know, the uncompression is called development. The uncompressing process is called neurodevelopment. We know a fair amount about that. There's a lot that we don't know. The particular sort of approach that we've taken so far exploits one aspect that we know is true in development, which is that to some extent the probability of connecting a pair of neurons depends on the identities of the pre and the postsynaptic neuron. But there's a lot of other stuff, and we certainly don't want to slavishly build all the detail. We're not trying to make a model. I'm not trying to make a model of development. I'm trying to get inspiration from it. But I think the other question you raised there, what you actually wrote was, is there any biological evidence for how the genome encodes or compresses the wiring? And that actually, we've cheated here by actually actively compressing information from a learned brain. Brains don't get to do that. Brains use, and biology uses an incredibly inefficient algorithm to encode the wiring diagram, and that algorithm is evolution. Evolution is basically a random walk through space without a gradient. And the only reason it works is that, I did a little back of the envelope calculation that suggests that contributing, prior to us, we have benefited from the existence of something like 10 to the 30 to 10 to the 40 other organisms. And only a small fraction of those survived and produced progeny. But that represents a huge amount of sort of wandering through this very, very high dimensional genomic space. So, no matter how fast the computers are, we can't match that. So basically the compression is done by wandering through genomic space, the uncompression through development. So the next question is, our goal, achieve mouse level AI. What time frame might this be achievable on? Well, so I'm actually working with a bunch of people to get a program going to actually set embodied AI as a goal. And, I don't think, obviously there's been a fair amount of work but it's been sort of haphazard. So I'm hoping that that if we actually go about it systematically, if we start by trying to achieve the capabilities of very simple organisms, and then actually we build on those capacities, if we build them into, if you like, a genome and use that as the starting point for the next step, I think it's actually not inconceivable. I'm thinking we could get to, in many ways, we could get to, you know, sort of mouse level AI within about 5 years, but it would take a serious concerted effort, and something I've been talking to colleagues about, and we're pretty interested in doing. Let's see. Interesting talk. Could you explain a bit more about how you perform the compression on the weight matrix? Yeah, so I mean, basically, well, we have a paper on that. I guess I would, which I think without sort of interactive feedback, it would be difficult to sort of get at what was unclear about my explanation. I can, I can put a link to that paper in the chat, but you could just, it's on bioRxiv. Hold on. Yeah, it's going to be hard. If you follow up with an email, I can give you a link to that paper, or you can look in bioRxiv for Zador and Kulikov. And it has some details. And if there are any questions, I'd be delighted to discuss them by email. Has machine learning standard practice independently discovered the genomic bottleneck? Specifically, I mean that ML common practice is creating a general language model with lots of training, then fine tuning to a specific task. Yes and no. That's certainly, it's certainly addressing some of the same problems. You know, the idea that, so the issue here is that these modern language models like GPT-3 and the ones beyond that are so expensive to train that, you know, only a handful of you know, industry players like Google and Facebook and OpenAI can afford to train them. I mean, they have an energy cost that, you know, causes the lights to dim in Boston for a couple of days. It's just, they're incredibly expensive. So rather than retraining the networks from scratch these days, people come up with these, I don't know, foundation models, they're sometimes called, and then the idea is that these can be further tweaked. So that does sort of address some of the same problem, but I think it still misses the beneficial effect of the genomic bottleneck, right? So the genomic bottleneck is a classic case of biology turning a bug into a feature, right? So, you know, the bug of the genome is that it's not big enough to store every connection. And so it's been forced to compress it. But you can think of that compression as acting as a regularizer that squeezes out the irrelevant information and discovers the relevant information. So I would argue that, you know, so in these foundational models, they just take the trained network with, you know, however 100 billion parameters or whatever it is, and hand that off, and that's the starting point for something else. I would say if you took that 100 billion parameter foundational model and squeezed it down to 100 million parameters or 100,000 parameters, then that would be capturing the full potential of the genomic bottleneck. Let's see. Are there any specific problems that you think the genomic bottleneck algorithm will be able to solve that current AI methods can't? Yeah. What we're really, so what I didn't have time to show you was that we get good transfer learning with the genomic bottleneck. So you train it, you train, for example, we train the cheetah on the standard cheetah environment. And then because it's convenient, we crank up the gravity in that environment by a factor of 2, or cut it down by a factor of 2. And if you just take the standard cheetah, it doesn't know how to move in that that new environment. Even if you take a standard transfer learning approach, it still doesn't learn how to move. But if you use the genomically compressed network, the cheetah on Jupiter quickly learns how to run. I think an even better application that we haven't really started pursuing, but I'm really excited about, is a task that's sometimes called something like continual learning. Not quite the same the way I envision it, but basically the real core of the genomic bottleneck is that, most modern networks are essentially idiot savants. They're very good at solving one thing, and then they don't know how to solve all these other things. Whereas what the genome does is it basically creates a hierarchical or quasi-hierarchical system that enables tremendous behavioral flexibility. So, there's a core component that is responsible for getting the limbs to move, right? And once you have an animal that can move, it should be able to move toward and away things. And then you combine that with, figuring out where it should move toward or away from. And it should be able to, switch rapidly from hunting to fleeing, all these different things. So, the way I see it is that what we would like to do is take a series of, let's say, 100 related tasks and build them into a genome. And the first one will take, I don't know, 1/100th of the total space of the original network. And then the second one might come close to doubling that. But by the time we add the 20th or 50th or 100th task, the increment in terms of what's needed in the genome to solve the 101st task should be absolutely minimal. And that's sort of the vision that I see. The real payoff that I see for this genomic bottleneck is the ability to to store all the information needed to solve a very, very large number of diverse tasks. And by the way, this is a weird sort of.
This is weird. Actually, Giancarlo, can you unmute yourself? I know you have a hand up. Now speak.
Okay. I was waiting for Tony to answer. So I thank you for this talk. I think you raised very valid points, and it's also nice to see a different point of view on these issues. But I want I have a feeling that though what the AI community, right, thinks about the intelligence that they want to understand is not so much how you learn to walk or even hunt, like a spider can do, but it's really how you learn to do math or play chess and things like that, right? I think this is what people think when they think about intelligence. In fact, human beings that go around in life and are discreetly successful who cannot understand math or appreciate Shakespeare, we don't consider them to be particularly intelligent. So the point I want to make here is that kind of intelligence does require, yes, a genome which is capable of producing the kind of beings that we are, with hands, with opposing thumbs, with eyes and everything. But it also takes a lifetime of learning so even, for example, to learn math from a textbook or with a teacher, right, which would be supervised learning or self-supervised learning, it takes years and years of training. And that requires, for example, the ability to speak. And we're not born with the ability to speak, right? A human infant uttered the first word after 2 years, despite the fact that he's been constantly bombarded with words spoken by other humans around the baby, right, for a couple of years. So I think that kind of intelligence, right, requires a lifetime of learning, which is on top of the innate structures that you may have. Even though, some innate structures are better than others. That's why we have Moses and Rafa Nadal as opposed, to other individuals. But these individuals still have to put in
right. So
thousands
I totally
of hours of training.
Yeah,
and that's the kind of training we want to, I think people want to understand artificial intelligence. That's the kind of learning they want to understand.
Right. So I totally get that's what people think they want. Okay.
Yeah.
I have a couple comments. The first is that, back in the day, right, what people thought they wanted was, and they thought would be really impressive and would be hard, right, was a computer that did the things that we find to be the pinnacle of intellectual achievement, like playing chess, right? And eventually, as you know, people discovered that playing chess is not the pinnacle of what we would call intellectual achievement by some measures, right? So the things that we thought were really hard, like playing chess, the things that, you know, were really impressive are not the things that computers that are ultimately that satisfying. I think the reason people focus on understanding chess and Shakespeare is that we take it for granted that by the time you get to appreciating Shakespeare and by the time you get to playing good chess, you've done all those other things that humans do sort of trivially, right? But that turns out to be false. So speaking of language in particular, you say that language is acquired, and that's true, but language is acquired, I think it's now pretty clear, only because we have innate circuitry that basically has a couple of slots open for the specifics of the language we acquire. So all humans are born with the innate ability to acquire a language, the language to which the language to which they're exposed. And the only thing we learn, it appears, are the details of the language that we're exposed to, the vocabulary of English, Italian, or Chinese, their syntax, the phonology of all these things. And so I think that the core thing is that if we want intelligent creatures that don't just mimic to some extent some of these, so I think some of these things will turn out to be non-mimicable. So even though language models are very impressive in some ways, they still don't understand in any meaningful sense what's going on. And no matter how big the language model is, it's very clear that they're not understanding in any meaningful sense. And I think that to get creatures that, or to get agents, artificial agents that actually do the things that we will really find, you know, mimic intelligence in ways that we find really satisfying, they'll have to in some way recapitulate the processes that we have all undergone. That's a belief. That's what the research program is. That we won't really know the answer to that until, you know, the singularity occurs in 2028, and we can find out exactly how we got to that singularity.
So that's true. And if I'm allowed another comment, but the thing is,
sure,
having those brain structures necessary to learn how to speak, they're a necessary condition to learn how to speak, not a sufficient condition, right? So you need to have the right stimulus for years.
Absolutely.
And the right procedures,
we know
right?
the language acquisition doesn't occur if you don't you don't put the child in the right environment.
Well, that's true for example, vision. You suture the eyes of an animal in the critical period of alphabet, they will never learn to speak. So I think that kind of, I'm only arguing that then of course what you learn is going to be encoded into the genome so that you can increase this infinite wisdom.
Well, actually, wait, just to be clear, what you learn isn't encoded in the genome. What you learn is encoded in the network circuitry enabled by the priors that are encoded in the genome. But yeah.
Yes, I express myself, you know, in a not precise way. But what I mean to say is that there's a kind of intelligence that is really given the basis, right? Which I don't know, an artificial neural network could be coded by how many layers you have, how many neurons in each layers, what kind of activation units, you know, all these kind of things, whether you have neuromodulators or not, whether you know, there's a lot of structure that is already there when you start making this model, trying to learn anything, of course, right? And I'm not saying nobody disputes that you need to start from, you know, I know you call it tabula rasa. It's not really a tabula rasa. There's already the wisdom of several generations of PhDs into those kind of models, right? The only thing that we're trying to do now is to learn the final step, which is really, okay, if I know English, can I learn French? Because I could be, you know what I mean? I could be, even my, you know, with a big brain and being, you know, going to French school, I may not be able to do that. Some people are better than others. And I think learning this kind of, you know, what we call the learning rule or the underlying, you know, mechanism is still, I think, something that relies almost exclusively on, you know, the daily experience.
Yeah, I mean, I think ultimately,
ultimately
this is, you know, the proof is going to be in the pudding. And I'm arguing that this is going to be a useful strategy. I'm going to try to hit a few of these other.
Yes. Maurice has his hand up. Maybe Maurice, quickly. Can you?
Sure. Thanks. This was a great talk. I really, it's pulled together a lot of ideas I've been thinking about for a long time. I'm a geneticist, not a mathematician by any stretch.
Okay.
But one thing that's always puzzled me, and is the idea of the genomic bottleneck. And it always the bit calculation. We can calculate the number in a genome easily, and that's why we do it. But it seems to be inadequate. And I put a comment up saying that what you neglect is the context of cellular structure, that there's so much what I call implicit information. Cellular structure.
And I think the answer
it seems,
to that is
although it seems that, of course, that all cells look alike, no matter how complex the organism.
Right.
But there may be some, I don't know, non-linearity in the way that interacts.
So I think the issue is this. If you just have DNA sitting in a bottle, in a vial, it doesn't make an organism, and it has to be in the right cellular environment. And the question is, how do we think about that cellular environment? The same is true. If I have a computer program that produces all the digits of pi, it still has to sit on some computer, right? And so, for the purposes of this discussion, right, I guess what I would say is that you need an enabling cell, right, one that's sufficiently rich to produce an animal without a brain. And so if I'm interested in how much information it takes to produce a brain, right, you can imagine, and again, because you're a geneticist, we can talk about the details of why this particular experiment won't work. But conceptually, right, I could take, let's leave mitochondria out of it, right, because they have their own genetic material. But I could take the DNA from my cell and try to coax a yeast into producing a mammalian cell. And if I got, it probably wouldn't work, and we could talk about why it wouldn't work. But I think conceptually, there's nothing fundamentally broken about the idea of slowly coaxing a yeast cell into producing a human cell. And so that tells you that essentially the key information, a yeast cell being a cell that, for those of you who are not biologists, yeast don't have brains. So whatever is important about the brain can't be present in the yeast. Certainly whatever is important about a wiring diagram can't be present in a yeast. So if you sort of grant me that in principle one could take the DNA from a human and put it in a yeast and try to boot up a human that way, I think that would be a better example, one that's a little bit more feasible maybe, is you could take the DNA from a human and put it into a rat. There are actually, that's something that still remains a little bit outside of what we can do, but there are trans-species, between pretty distant species, trans-species DNA transfer that actually has worked. And so whatever's smarter about us compared with a rat, we could get at by taking the DNA from a human and putting it into a rat cell. So I think that conceptually it's not, one has to be a little bit careful, but I think you get the right intuition from thinking about taking the DNA from an organism with a complex brain and putting it into the cellular host of an animal that has little or no brain. That would be my answer to that.
Susan, you want to speak or unmute yourself? There you go.
So this is a great talk. Thank you so much. I was just thinking, I study language and language acquisition and language use. So if you truly wanted to have a language learning ability in AI, I think you would need to have the ability to understand interaction with another being that's contingent. Certainly humans can't learn a language or acquire a language in any natural way without interaction without other humans that's contingent. Children can't learn language from watching TV, et cetera. So it doesn't seem to be the case that any of my colleagues in NLP are looking at interaction or the sort of knowledge that's encoded in the genomic bottleneck that has interactive intelligence.
I couldn't agree with you more. In fact, for a while, it's a back burner project that I started working on with Alex, and it just sort of, it's a little bit beyond what we can do right now, is to actually try to, so I believe that language is for, interacting, as you say, and that, my just so story about the evolution of language, whether it's true or not, I find sort of inspirational, is the idea that in the beginning, we lived in small groups, and what language began as the imperative, right? You go there, I go here, and that allowed us to cooperate in hunting, right? So sort of starting from that, we sort of envisioned creating a virtual world in which we had interacting agents that where one had information about the world that it needed to convey to the other so that they would jointly optimize their success. So, you might have agents in a simple environment that went looking for things, went hunting together. And we kind of thought that what we would end up doing is producing, they would be allowed to transmit information to each other, transmit bit strings to each other, and that they would jointly evolve the ability to interpret those bit strings. And we wanted to evolve language in this. I still think it's a cool idea. There has been work that's kind of like that. We sort of backburnered it because it would be, it would take a serious effort, and we just didn't have the resources to pursue it. But I completely agree with you that what we would need to generate language is agents interacting in a world where the interactions were, where words had meaning, where the interacting strings had semantics in the form of driving particular actions.
Right. And even before that, before being intelligent about interaction, agents need to understand, they need to recognize intention from other agents. So you can do a whole lot non-verbally with where someone's looking or whatever, and animals do this. Yeah. Some animals do this as well.
Yeah. So I completely agree. I'd be interested in talking to you more about this idea.
I'll confess I have a question. How deterministic is kind of the brain development in a higher organism? So you told me that kind of two C. elegans, you cut them up, they're the same. To what extent am I like, Maurice or my clone?
Yeah. So the short answer is we can't really know for sure. But the answer is we kind of do know anyway. So in very simple organisms like C. elegans, right, everything is extremely stereotyped. There are 302 neurons, and people who study C. elegans have names for each of them, right? So I don't know what those names are, but we could, they're unique. You can return to them. They have the same position, the same gene expression, et cetera, et cetera. As you go up to slightly more complex organisms like Drosophila, or I don't know, slightly, as you go up to more complex organisms like Drosophila, which a fruit fly, which has about 100,000 neurons, there are definitely some neurons that have that property. They're stereotyped. They come in pairs, right? People who study Drosophila will say that there's, the such and such neuron on the left side and the such and such neuron on the right side. But there are other neurons that appear to be sort of a little bit more stochastic, right? And then by the time you get up to humans, there's just a whole lot of stochasticity in development. That said, right, the brains, we believe that the brains of genetically identical organisms are more similar, all other things being equal, than the brains of genetically different organisms. So basically, the rules as you get up, the developmental rules as you get up to mammals and certainly to humans have a large stochastic component. The rule might take the form of, there is a cell type X, and with probability, A, it produces a connection to another nearby cell of cell type Y, but only if cell type Y doesn't already have a lot of connections from other cells of cell type A, right? That's the kind of stochastic rule that would be formed, right? And so that means that the brains are going to be, similar, right? We know that, basic brain structures appear in the same places within a species and even across species, but the details will, you can think about a particular wiring diagram as being drawn from some very, hard to specify probability distribution, one that's induced by these stochastic developmental rules.
Okay, good. Are there any other questions? Oh, I think Dakota has a question. Dakota, do you wish to speak?
Sure, yeah, I just posted a doozy. I'll give you a second to read that one.
What is the extent of the transfer learning experiments? Has anything been attempted which observes what happens when the genomic cookbook is no longer directly relevant to the given phenotype, i.e. rewiring after the removal of previously functional regions as a result of the stroke, or trying to get your cheetah to relearn how to move after its leg? So I can't tell if you're asking about biology or about our very, very simple model.
Sort of a little bit of both. I suppose I conflated both in the question, but my idea would be that there has to be some line where this genomic bottleneck starts to break down, where like their compressed information isn't useful to the current situation. I'm wondering if that could happen given a change in the phenotype. So you still have the same cookbook, but suddenly like it doesn't make sense. You don't have these ingredients at home. You can't even try to follow.
Oh, that's an interesting way to look at it. So I'll say that, you know, I'll start with the easy one. We're still early days in the genomic bottleneck approach, and it breaks down very quickly. I mean, you know, we're pleased that we get some compression. We're pleased that we get some transfer learning, but it's nowhere near as flexible as, you know, we would like it to be, much less compared with biology. The next question about how flexible is biology is a little bit more complicated, especially because there are a couple questions mixed in there. You mentioned a stroke. Well, stroke is limited not just by the flexibility of, you know, the developmental program, but by the fact that the way development works, a lot of these sort of developmental processes basically shut down at some point, right? So it turns out that so, you know, the reason you can grow your arm once, but if you chop it off, it's hard to grow it a second time, right? If you remove some of the ingredients that might have been useful for growing your arm the first time, you still often will get something like an arm. But if you remove those after the critical period in development has shut off, you will it's very hard to get it back. That's why sort of adult regeneration is more complicated. So that's why adult regeneration. Yeah, I thought I heard something on my side.
Yeah. Okay, I think I think this was a very interesting talk, and I think other people will agree with me. So let's thank our speaker, and let's say hopefully this is the beginning of a long and
yeah. Thank you all,
friendship.
and I'd love to, you know, I'm excited to keep talking about these things and getting different perspectives. So I'd be delighted to, you know, follow this up in email or something else.
Okay, sounds
Thanks so much.
Thank you very much.
Thank you.
Okay, goodbye, y'all.